Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Safety and Tolerability in Oncology Trials, Cardiac Imaging, ECG, and the Role for Patient-Reported Outcomes. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the richest clinical evidence in the industry for their pharmaceutical, biotech, and medical device partners. Across decentralized hybrid and site-based trials, their deep scientific expertise, global scale, and the broadest endpoint technology platform in the industry allows their partners to transform lives. With 11 facilities in 6 countries, Clario's global team of science, technology, and operational experts has helped deliver over 21,000 trials and 870 regulatory approvals for over 5 million patients in 120 countries. Now, it's my pleasure to introduce you to our moderator for today's webinar. As Clario's Chief Medical Officer, Dr. Todd Rudeau, there he is, provides medical and scientific leadership across the organization. Passionate about leveraging their scientific expertise to support their customer success, Dr. Rudeau has focused efforts on optimizing their solutions across the therapeutic areas. His team supports Clario's clients by providing expert consulting services on scientific and regulatory strategy and ensures their product portfolio is scientifically robust, generating high-quality data to support clinical trial endpoints. With nearly 20 years of clinical cardiology and pharmaceutical research experience, Dr. Rudeau has his career predominantly on drug safety. So there he is. So when you're ready, Todd, he is going to introduce you to our speakers for today's webinar.
Okay, great. Thanks, Sonia, for the very kind introduction. And thanks to all of you who have taken the time to join us today. We're glad to see such enthusiasm and interest in this session as we discuss safety and tolerability in oncology trials, with the focus specifically on cardiac safety and the role for patient-reported outcomes. Joining me today are Dr. Bob Kleiman, Dr. Maury Krantz, and Dr. Kelly Dumais. Dr. Kleiman is the Vice President and Chief Medical Officer for Cardiology at Clario. He's board certified in cardiovascular medicine and cardiac electrophysiology, trained at the University of Pennsylvania here in Philadelphia, practiced for about 12 years before joining Clario in 2003. He oversees all of Clario's cardiology services, including the ECG Core Lab, as well as cardiac safety consulting services for sponsors and the statistical analysis and expert report writing teams. Dr. Krantz is a senior cardiologist and chief scientific advisor of cardiac safety and cardiovascular imaging at Clario, and has board certifications in cardiovascular medicine and echocardiography. He's the governor of the Colorado chapter of the American College of Cardiology, serves on the board of trustees for the International Society of Holter and Non-Invasive Electrocardiography, and in his current role provides consultative support to sponsors on optimizing their cardiac safety strategy with a particular focus on cardiovascular imaging, and this is where I'll concentrate for today for us. And Dr. Dumais is a behavioral neuroscientist by training, serves as principal scientific advisor on the ECOA science team at Clario. She has deep expertise in the implementation of electronic outcomes assessments in clinical studies and the development and validation of patient-reported outcomes to support regulatory submission. She also has expertise in development of custom site rater training and participant training to improve data accuracy. And today we'll share an overview on the evolving role of patient-reported outcomes in oncology drug development. We can go to the next slide, please. To help set the stage for today's webinar, I just wanted to highlight what makes oncology drug development such a unique space. Many of the considerations listed here are unique to oncology in particular, and therefore present a deviation from the more typical or established norms of drug development. Just to highlight a few, in oncology development programs, rarely is a healthy volunteer phase included, nor is a placebo comparator, so confounding factors including significant comorbidities, are present which make data interpretation challenging. In addition, the development programs can include long extension studies, even extensions directly from phase 1, and the regulatory pathways for approval are quite different as well. The patients themselves are unique in that they are often heavily pretreated, having already failed first and second line regimens, particularly in early phase oncology development. And in such sick populations, the adverse event rates are much higher, regardless of any treatment-related risks. Any proposals, therefore, to add further assessments within a protocol need to be balanced against the consideration for increasing patient burden. And while that's a true statement for any therapy area, it's particularly a concern when treating sick cancer patients in such a frail state. And lastly, the drugs themselves. In many cases, they're more toxic than those in other therapy areas, yet the tolerance for that toxicity from a benefit-risk perspective is quite different. We're now, however, seeing a shift towards more targeted therapies that have reduced toxicity, but also are far more selective. And while generally that's advantageous, it can be a challenge operationally now to find patients and efficiently enroll, as sponsors are seeking only a small subset of patients with a particular cancer type or genetic signature. So with that said, I will hand over now to Dr. Kleiman to take us through the key considerations when assessing cardiac safety in an oncology development program, with a focus on the ECG and blood pressure testing in particular. Bob.
Thanks, Todd. Hi, folks. Thanks for joining us. I'm Bob Kleiman, and I'm going to talk about cardiac safety as it pertains to oncology trials.
Just need to reshare your slides.
I hope you can see the slides now. So first, one has to test the cardiac safety for all new drugs, and it's really not a complicated matter. All drugs, regardless of the route of administration, are delivered through the bloodstream, and that means that the heart and cardiac myocytes are exposed to any drug that we administer, other than perhaps some topicals that aren't absorbed, some unusual GI drugs. So cardiac toxicities are possible for any new drug until they've been excluded in clinical trials. So I made this slide up a few years ago, but it makes the point that I'd like everyone to see. This is a list of all of the oncology drugs that were approved in 2016 and 2017, and there are quite a few in multiple different classes. And here I've highlighted those that have cardiovascular toxicities mentioned in the label. And as you can see, it's more than half of them. This is probably the complication we worry about the most. This is, of course, torsades de pointes, a form of polymorphic ventricular tachycardia that often leads to cardiac arrest and sudden death. Now, as I've said, a lot of oncologic drugs have cardiac toxicities. But remember, the regulator's job is to weigh risks and benefits. And in oncology, that balance is really shifted towards benefit. Side effects are expected for most drugs, and in general can be managed. But on the other hand, there really isn't any benefit to bringing someone's cancer into remission if they die of sudden death as a result of the treatment. And it's hard to balance having an improvement in your malignancy and gaining a few extra months of life if it's at the expense of heart failure that's so severe that a patient can't walk 20 feet without getting short of breath. And more, the FDA and other regulators have been looking not just at objective endpoints, but also at quality of life and patient-reported outcomes to help understand what is the true benefit for the patient rather than what is a statistical benefit. best example of this is the poster child for a toxic oncologic, arsenic trioxide. Yes, as in arsenic and old lace. Arsenic is obviously a poison. And the efficacy for treating certain unusual leukemias was established well before anyone recognized the cardiovascular risk. But when we went back and looked at the ECGs that had been collected in clinical trials, it became obvious that arsenic trioxide produces enormous QT prolongation, more than probably any other non-antiarrhythmic drug. Yet it's approved. And the reason is, with correct screening of patients with proper monitoring, it can be used relatively safely. So the current guidances are that you really have to assess the ECG effects of any new drug, oncology or otherwise, but it's less about approval of the drug than about informing the regulators how to write a label that helps prescribers use the drug safely. And again, this is usually just a labeling issue. Note that even some drugs with very large cardiovascular toxicities, arsenic, Herceptin, some TKIs, are approved and widely used. Again, it's not about approval or lack of approval. It's about how do we write a label for a new drug. In terms of QTc, which is probably the cardiovascular toxicity that comes to mind most rapidly, we like, if possible, to assess ECG effects, particularly QTc, in the first-in-human phase 1/2 studies. The reason is, to do this, we need intense time-matched ECGs and PK. We often collect the ECGs with a continuous 12-lead recorder. And phase 1/2 studies, particularly dose escalation portions, are fantastic because they provide the widest range of exposures you'll have in any study, and sometimes the highest doses as well. And that is perfect for concentration QTc analysis. Later trials may allow you to collect ECGs relatively simply, but collecting the additional PK time points, that can be a challenge in a phase 3 study. So ideally, we'd like to see this in the first-in-human trials. And again, it's not to exclude a small effect, as we might do for a new antihistamine, a new antimicrobial. The goal is to determine, is there a large QTc effect, meaning greater than 20 milliseconds, so that a label can be written to inform prescribers how to use it safely. Now, ECGs are collected in most oncology trials, regardless of the intent, whether it's for QT assessment or just for safety purposes. But I'm often asked, is there an advantage to centralizing the ECGs in the oncology studies? So to answer that, we did a study where we looked at about 270,000 consecutive ECGs, all recorded from oncology studies, for which we had both the ECG machine algorithm measurements and our centralized measurements. It's about 18,000 subjects across almost 300 studies. And what we found is that ECG machines produce a lot of false positives. So for instance, when the ECG machine reports a QTCF of greater than 500 milliseconds, there's a more than 75% likelihood that that is a false positive, with a mean difference of about 80 milliseconds. So that means if the machine reports a QTc of 520 milliseconds, it's a rather frightening value and would probably lead to exclusion or termination of treatment, or at least withholding doses. There's a good chance that the QTc not only is below 500 milliseconds, but actually may be in the completely normal range. And that has important implications both for recruiting for studies as well as for the conduct of the studies. Similarly, there are many false negatives, which are even more frightening because those are difficult to pick up. So again, we recommend centralization of the ECGs, both for safety purposes as well as for collecting data for submission purposes. Now, although I'm an electrophysiologist and QTC is my particular passion, there are a lot of non-QTC related cardiac toxicities. Some are detected on the ECG or may produce non-specific ECG changes, but others really need to be picked up with a different modality. For instance, heart failure or pericarditis are typically best picked up with echocardiography. Arrhythmias, bradycardias, tachycardias, atrial fibrillation, heart block are usually better picked up with continuous recordings with a Holter or a long-term patch. And of course, as we all know, the FDA is increasingly interested in drugs producing blood pressure increases, and that's best picked up with ambulatory blood pressure measurement or with high-quality clinic or home blood pressure assessments. We actually have a couple of different complementary methods for measuring blood pressure in clinical trials. The best is probably ambulatory blood pressure measurement at this time, and we can get a full assessment of the blood pressure effect of a drug over 24 hours. In contrast, clinic blood pressure measuring can be very precise, but it only measures blood pressure at the time that the patient is actually in the clinic and may not have much relationship to the dosing time. And more recently, we started doing home blood pressure monitoring by providing uniform devices for all the patients and centrally acquiring the blood pressure data. And that can be customized to have a patient record their blood pressure once a week or 3 times a day, as often or as infrequently as you'd like. Quickly, just a couple of examples. VEGF inhibitors are a class with many cardiovascular complications and are used in many malignancies. Cardiac side effects can include hypertension, sometimes as severe as hypertensive emergency, left ventricular dysfunction, microvasculopathy, QTC prolongation. Similarly, tyrosine kinase inhibitors are all over the globe now, very many already approved and many in development. And many tyrosine kinases are expressed in cardiac and vascular tissue. So the side effects can again include hypertension, left ventricular dysfunction, QTC prolongation, thrombosis, myocardial ischemia, even myocardial infarction. So to close my segment, the current best practices for cardiac safety assessments during oncology studies are first to collect ECGs during phase 1 through phase 3, at least for basic monitoring. Many of these patients are older, have been pre-treated with multiple rounds of therapy, have con meds, and may have underlying cardiac disease. We often see clinical assessment of cardiac troponin or BNP. These are inexpensive, easy to obtain, and they may detect cardiac toxicity well before clinical symptoms of heart failure develop or even before anything shows up on the imaging studies, which may allow prevention of permanent left ventricular damage. As I mentioned, blood pressure assessments are becoming increasingly important. And finally, we often see serial assessments of left ventricular function by echo, MRI, occasionally still by MUGA scan. And with that, I'm going to turn the stage over to Dr. Krantz, who's going to tell us more about cardiovascular imaging. Thanks.
Thanks, Bob. I really appreciate it. That was a tour de force. Really like that introduction. So yeah, my name is Maury. I'll be focusing mainly on cardiovascular imaging in oncology, and I serve on the Clario Cardio Oncology Work Group, which is a nice collaboration between oncologists, cardiologists, and other preventive physicians and clinicians. So our goals today will look beyond the ECG work and the blood pressure changes that are rigorously measured centrally that Dr. Feynman alluded to, but also look at myocardial toxicity. And the primary focus, as you'll hear over and over, will be the main pumping chamber of the heart or the left ventricle. But I will add that the pericardium, as well as the myocardium itself, can be important as well. The myocardium from the tyrosine kinase inhibitors, for example, can get inflamed, and that can lead to heart damage. But the pericardium, the sac around the heart, can also become inflamed, and that can lead to fluid accumulation and ultimately pressure and strangulation of the heart, which we call cardiac tamponade. And although rare, is obviously a life-threatening emergency. So what I'm going to do, which is a little bit unorthodox, is talk about cardio-oncology through the lens of a disease-specific case of a patient who has a multiple myeloma or a plasmacytoma and developed cardiac amyloidosis, which is a systemic disease. And we'll measure toxicity, health status, and how we actually can treat this itself. Of course, Dr. Kleinman mentioned the risk-based approach. There's both the drug class itself and how it can affect the heart, but also the patient risk. If there's multiple comorbidities, you can imagine that patient would be much more susceptible. I'll try to allude to some best practices and explore some of the inherent linkages between heart function and patient-reported outcomes, which we'll discuss later. So what are the issues at hand? Obviously, the emerging targeted therapies are better, as you heard, but it's hard sometimes to differentiate between a cancer cell and a non-cancer cell. And this can be leading to the things we discussed: myocarditis, pericardial inflammation, and of course, left ventricular dysfunction. So I won't go into a lot of detail about the mechanisms of how the heart itself is damaged. Suffice it to say, there are numerous mechanisms, including oxidative stress from free radicals, mitochondria damage or changes in energetics, and you can see some examples here, and increasingly immunologic compounds such as monoclonal antibodies, the checkpoint inhibitors, et cetera. And one thing that is critical when we look at a program where we're helping sponsors for development, we look at the drug itself, its preclinical work, what clinical data we have, but also if it's combined with another drug, does that increase the cardiotoxic risk? And that is really an important issue to deal with. So I think to me, the million-dollar question for everyone is, what is the classic manifestation of myocardial injury after chemo? And it's pretty simple. I'm not sure how well these slides are playing on your end, but basically it can affect the left ventricle. Just to orient you, if you look at the left panel, the left ventricle is the pumping chamber on the right ventricle on the left, and you see the atria below where my cursor is, if you can see that, but the heart is pumping relatively normally. By contrast, if you look at the right panel, you can see that the heart is actually dilated and really only moving very little. And this clinically, as Dr. Kleinman alluded to, can result in congestive heart failure, symptoms of dyspnea, poor effort intolerance, and he gave the example of someone who could only walk 20 feet. So in addition to the heart moving in and out, I want to bring up a little topic that's kind of interesting, and that's the notion of strain imaging. On the middle panel, you can see these little speckles, blue and orange dots, and there's a way to look at the heart motion that's more intensive than just looking at the endocardial thickening. So the heart doesn't just move in one dimension, but it's sort of like a wet rag. If you can wring it and get the water out, that can really show different aspects of the cardiac performance. And why is this important? Well, if you look at this function or global longitudinal strain, and I'm not sure how well this is coming to you, but the idea is that rather than looking at one dimension, the inward and outward, if you look at longitudinal sliding of the heart, clockwise rotation, and even counterclockwise rotation, you can see much more subtle changes and recognize toxicity early before this becomes manifest clinically and the patient actually begins to feel bad and suffer. And that's sort of depicted by these arrows that are sort of coming out of the heart and moving in multiple directions on the image to your right. So if you look at the continuum of myocardial toxicity as a echocardiographer, the way I look at it is the first thing that happens as there's damage to the heart is it becomes stiffer. It doesn't relax well. That's the panel on the left. Then you can see impaired global longitudinal strain, which is actually a systolic marker, but preclinical. And this can happen before the ejection fraction actually is fully reduced. So I think that's an important marker. Go to the next slide here. It'll work. Okay. Let's see here. People can see you. Okay. There we go. So when do we screen a patient? So I think the idea that Bob mentioned was that it's really the notion of a risk-based approach. How sick is the patient and how toxic is the drug? As you can see on the left, using the example of an anti-human epidermal growth factor inhibitor, you will look at the electrocardiogram, certainly two-dimensional echo, both for low-risk and high-risk patients. But as Bob mentioned, also integral to the assessment of cardiac safety is the cardiac troponin and the brain natriuretic peptide. And I think that's really important to measure these things, because these are measured in the billionth of gram range and can detect very early dysfunction. So we mentioned the echo. If you look at the bottom in terms of the recommendations from the 2022 European Society Guidelines for Cardio-Oncology, echo is first-line modality. There's a movement towards 3D, which itself has some challenges, as you can imagine. But GLS, the strain that I showed you earlier, is now recommended in all patients if available. So let's start with the case. So my patient is a 77-year-old man who has shortness of breath, suggesting heart failure. He has weakness, dizziness, and even is falling down periodically, having syncope. He has kidney failure, and when we do a urinalysis, it shows some light change, or these proteins that are suggestive of amyloidosis, which is confirmed by biopsy. So let's look at an echo for this patient. If you look at the left panel, what you can see is where the white arrows, there's almost a lot of bright white dots in the ultrasound of the left ventricle. And this is sort of a sparkling appearance that we call the Vincent van Gogh properties. And this is demonstrated on the top right of your screen, where you can see that the amyloid proteins on the top right are actually depositing and intercalating within the myocardium and damaging the cells. So on transthoracic echo, we did an echo, and we actually went ahead and did the strain map, and that's on your left. And you see this sort of classic cherry on top. If you look at this map, in the middle, there's a red dot that almost looks like the cherry on top of an ice cream sundae. And that depicts the fact that the upper part of the heart is moving very well, but the basilar function is not moving at all. There's a much more sensitive and specific test we can do, which is called a cardiac MRI. And that's shown on the right of your screen, where you can see a patient with amyloidosis on the left and the healthy comparison. Clearly, the walls are much thicker with amyloidosis, with a change in T1 time in the mapping, and actually the presence of scar tissue from those amyloid fibrils, which shows up as sort of a white patchy appearance in the tomographic image of the heart as you see it. So we mentioned strain, and I think what I like about this new marker is that it's actually more sensitive in predicting the risk of heart failure or death, which is shown on the y-axis. And you can see that strain parameters are in the 90s to 100%, whereas the classic ejection fraction on the far right of your screen is only about 34% predicted. So I think compared with conventional parameters such as the ECG that Bob mentioned and the ejection fraction, we might be able to have additional prognostic information from this value. So let's get back to our patient. So what did we do? I will do a full disclosure. I am not an oncologist, so I will only say sideboard D plus D. I can barely pronounce cyclophosphamide, but also the patient received a proteasome inhibitor, steroid, and monoclonal antibody. And what you can see for cardiac amyloidosis is, of course, a clinical assessment is demanded in all patients, but blood pressure and ECG are central, and even home blood pressure monitoring. We mentioned the blood tests, the natriuretic peptides and troponin, but so certainly the TTE and CMR for diagnosis, but that ultimately sequential measures of TTE or transthoracic echo should be performed relatively frequently as part of your development program and even in clinical practice. One question I want to bring up is, this is sort of a safety issue, is it not? But what about efficacy when we look at a malignancy such as cardiac amyloidosis? And I wanted to just share one study that was published in the European Heart Journal a year or so ago, and looking at a different type of amyloidosis, not like the patient I described with AL or plasmacytoma, but this is transthyretin amyloid, where these tetramers start to aggregate and intercalate into the heart. They normally carry thyroid hormone and vitamin A through the bloodstream, but they become toxic to the heart. And again, you can see in the middle of the screen that cherry on top appearance of your global strain map. What's interesting is if you look at the bar graph on the right with the box and whisker plots, the absolute change in strain, it appears to get worse to a greater extent with the red and better with the blue, with the tafamidis. So I think seeing lesser deterioration in the global strain and myocardial work may suggest that we can actually not just keep our patients safe, but maybe improve them with targeted therapies. So putting it all together, I'd first like to thank everyone for taking time to join us today for this brainstorming session. But I think it's really about an integrative approach to research services. And I think systemic amyloidosis is a beautiful example. Certainly on the left, we're going to look for imaging. We're going to see if cancers are improving. We're going to check the light chains. We're going to obviously look at blood pressure and ECG. But also we're going to use cardiac imaging to assess safety and in certain instances, maybe even efficacy. But I think perhaps the most important aspect of efficacy is patient-related outcomes. At the end of the day, from a regulatory perspective, it's how you feel and how you function that really wins the day. And I think we have eCOA services like most central lab where we look at electronic clinical outcome assessments, and we can even look at precision motion and see if patients are falling down and having syncope spells. So with that, I'll turn it over to Kelly, who will give us some great information on patient-reported outcome. I thank everyone for their time.
Thank you very much, Maury. Hi, I'm Kelly Dumais. I'm a principal scientific advisor here at Clario, and yes, I'm very excited to be talking to you today about patient-reported outcomes and the important role that they play in this discussion. So I'll start really by jumping right in and answering the question of why are we talking about patient-reported outcomes in a webinar about safety and tolerability in oncology trials? A couple of months ago, the FDA released this draft guidance shown here that does call out the use of PROs, patient-reported outcomes, for the assessment of tolerability in oncology trials. This guidance alone is not the reason we're here talking about PROs for tolerability assessments. My portion of this talk will review, lots of reasons why we're including PROs in this discussion, but this guidance is, I think, a good place to start. So the guidance does state patient-reported outcomes can provide a systematic and quantitative assessment of expected symptomatic adverse events and their impact on function. Inclusion of PROs should be considered to enhance the assessment of tolerability in early phase dosage finding trials. And then they recommend to go to the draft core PROs and oncology guidance that the FDA put out a couple of years ago. So I just wanted to take a minute to break this down for a minute. So systematic and quantitative assessment. This is what the FDA has been emphasizing for a while now when it comes to evaluating the patient experience. How can we systematically quantify the impact of a treatment on patients' health? And using PROs is really one way to accomplish this. Impact on function. Having objective measures like blood pressure, cardiotoxicity, is undoubtedly important, but they don't say anything about how the patient feels, right? Or how it impacts their ability to work or do daily activities. So the impact that these other effects has on functioning, that's something really important to evaluate, and that's something that PROs can answer. And then enhance the assessment of tolerability, right? We're not looking for PROs to replace any other method of safety and tolerability assessment. It's a way to provide deeper insights into how tolerable a drug is from the patient's perspective. So it's almost shocking to think we've come this far without actually directly asking patients and having them report themselves about their subjective symptomatic experiences and how it impacts their function. And lastly, that early phase bit. That's maybe an obvious call out, but it's really these early phase studies in oncology where you might get the best benefit of using PROs to enhance the assessment of tolerability. And then also, obviously, PROs will enhance assessment of efficacy as well, which we'll talk about too. So today, what I'm really hoping to do is provide kind of the history of how the FDA came to this conclusion that PROs are also an important aspect to include in oncology clinical trials, both for the measurement of efficacy, but also for the enhancement on the assessment of tolerability as well. So let's take a step back and take a look at, why there has been this growing interest in using PROs generally in oncology clinical trials. And the first and maybe most obvious is that patients should and want to know how they will feel in response to taking an oncology treatment, right? So cancer survival rates are increasing, which is an appreciated accumulation of all of the medical advancements we've made, but it also means that more people now are living with cancer more than ever before. So it's not just about survival. We really want to make sure we're thinking about how patients are living with cancer. And I'm not suggesting that symptoms are more or less important than survival. They're not to be compared, but the reason why PROs are so important is because we really owe it to patients to take their symptoms and experiences seriously and therefore measure them in a systematic way. And also so that we'll be able to tell them how the treatment will make them feel so that they can prepare as they're about to embark on a new treatment, or also just have all of the information they need to make informed decisions about their treatment in a world where there are so many increasing options for types of treatments, which then leads to number two, the competitive market landscape. Interest in PROs is growing partly because sponsors are starting to see that this type of patient experience information when it's collected in a systematic way, could be one way that they could differentiate their drug on the market. And then finally, why is interest in PROS growing? It goes back to the regulatory agencies. So both FDA and EMA have made it very clear that the use of PROS in oncology trials are important and necessary in order to provide kind of that full picture of the clinical benefit and to inform risk-benefit evaluations as well. So let's take a minute to kind of try to look at the big picture of the oncology regulatory landscape in recent years. And here you can clearly see this increased attention on PROs from both the FDA and EMA. There are lots of other oncology guidances not included here. This is just a snapshot of those that relate or mention PROs specifically. So the EMA was actually the first to come out with a PRO guidance specifically for oncology, which they released as an appendix 2 to their oncology guidance in 2016. And of course, the FDA released their guidance on PROs in oncology in 2021, a couple of years ago. Both EMA and FDA have also stated the importance of PROs in their general oncology guidances, which were updated in 2018 and 2020. So you can really see this, explosion in just the past few years of the emphasis on PROs. And then, of course, that dose optimization guidance that references PROs there. And then scattered within here are also numerous FDA initiatives that have come out of their Oncology Center of Excellence group, all of which are not listed here. But one I did mention was the Project Patient Voice, because this was something that was launched in 2020. It's an online platform for patients and their healthcare providers to actually see patient-reported symptom data that was collected as part of oncology clinical trials that maybe were not on the label, but they're providing their patient-reported outcome data so that they can see this and make more informed decisions, as I said, about their treatment plans. So that's a great program that the FDA launched, and I encourage you to look at that further into that if you're interested. The FDA has also launched an annual workshop on clinical outcome assessments focusing on PROs in cancer clinical trials. So every year they have a discussion on different PRO topics and as they relate to oncology trials. Last year was the use of PROs in open label trials. This year they'll actually be focusing on side effect impacts, so extremely relevant to today's topic. So I encourage you also to go look at that workshop if you're interested. It's in June. They're free public virtual events. So if you're interested, go check that out. So I'll next dive a little bit deeper into that core PRO guidance from the FDA in a bit more detail, because the FDA has done a really great job in really outlining the key PROs that are important for assessing efficacy and even enhanced assessment of tolerability in oncology trials. So the 5 core PRO categories that they recommend to measure are disease-related symptoms, physical function, and role function. Those 2 are commonly fulfilled by using for example, EROTC QLQC 30, which measures patients' ability to perform physical functions and daily activities. And then there are the symptomatic adverse events and the overall side effect impact summary measure. These are, most commonly fulfilled by the PRO CTCAE, which I'll discuss in more detail in a few slides, and then the FACT GP5 or other similar global item that measures just a one question measure of how bothered patients are by the side effects of their symptoms. Another important thing to mention is the frequency for which they recommend to collect PROs, because they also really emphasize that in this guidance. So traditionally, if PROs are included in an oncology study, they're typically collected at site visits only, maybe the first day of some of the early treatment cycles. But the problem with that is that these PROs typically have a 7-day recall period. So that means if you collect the PRO at the start of, say, a treatment cycle, you're only actually collecting the patient's experience from the last 7 days of the previous treatment cycle. And we know the intensity of toxicity will really vary depending on, say, the type of drug. Is it IV? Is it oral? So you might either be overestimating or underestimating the side effects that they're experiencing by not looking at that whole picture. So as a result, the FDA now recommends more frequent data collection, giving an example PRO frequency table there. by which they even recommend weekly assessment of some PROs during the first, couple of months, the first few treatment cycles, and then maybe monthly after that, and then maybe 3 months after that. So generally, FDA is really looking for more thorough evaluation of patient symptoms and quality of life, and then also more frequent assessments as well. So to answer the question of, when do you incorporate these PROs, it would be as early as possible. And this is according to an FDA representative speaking at last year's Oncology Public Workshop on COAs. There are a number of reasons why incorporating PROs early in drug development programs is beneficial. The first is that it's really truly the patient-centric approach, right? So with early phase being the first opportunity to formally include that patient voice via PROs, the emphasis on patient centricity by the FDA, as we see, is only growing. In fact, they just released their 4th guidance on patient-focused drug development series last week or the week before, not oncology focused, but just general across all TAs. They have 4 guidance series of, how to really make sure that your studies are patient focused. It's also a way to optimize later phase or phase 3 pro strategy. So if you're including pros at early phase, you can explore, different endpoints, different assessments, measure and discover what's really important to patients. And then if you collect pros early, you can then use that information to base your phase 3 strategy on, understand your focus areas, what might be differentiating your drug, as I mentioned, in terms of that patient symptoms and experiences. It also helps you to be submission ready, right? So expectations from the FDA is that you will be incorporating PROs, and many drug approvals are based on those early phase, non-phase 3 trials. So this really warrants early incorporation of PROs to make sure that you're aligned with regulatory recommendations. And then finally, incorporating PROs early allows you to enhance evaluation of tolerability, and that's why we're discussing it today. So optimal dose finding might include this evidence of patient tolerability using these self-report measures. So now that we've established kind of the importance of early phase PROs, let's deep dive into what PRO tools should you be using, especially focusing here on the tolerability aspect. So as mentioned, one of the most common ones to assess symptomatic side effects would be the PRO CTCAE, which measures patient-reported symptomatic adverse events. So currently, safety is mainly based on clinician evaluation of adverse events and is reported using the clinician-reported CTCAE, which is that grading system used across all cancer clinical trials. The CTCAE data includes both symptomatic adverse events and laboratory and clinical adverse events as well. So safety and tolerability, though they're related, they're distinct from one another, right? Such that safety reflects the medical risk to the patient, frequently involving clinical judgment, and tolerability reflects the extent to which adverse events impact the patient's willingness to remain on treatment, right? And so the key contributor to tolerability includes those side effects that are symptomatic and maybe bothersome to the patient, as compared with the laboratory abnormalities that might go unnoticed by patients. So although the assessment of safety requires clinical judgment, relying on clinical assessments of the patients at CTCAE, the ability to continue a treatment at its recommended dose of tolerability could be better informed by the patient's assessment of symptoms and their side effects and their impact on their function, as I mentioned. So therefore, the National Cancer Institute developed a PRO version of the CTCAE titled PRO CTCAE. Important to note again that here, the PRO CTCAE is a complementary tool for the purpose of enhanced tolerability assessment. So this does not replace the clinician-reported CTCAE. The clinician CTCAE remains the core of safety monitoring, right? The PRO data without clinical interpretation are considered the safety data. So the PRO CTCAE can just add that complementary information to make sure that we're incorporating, again, the impact of these symptomatic adverse events from the patient's perspective, because we know that these symptomatic adverse events like pain and nausea fatigue, these are really best interpreted and best measured by the patient themselves. So next, I'll show you some data on, why measuring these symptomatic AEs from patients, as I mentioned, and not just clinicians, can be important. So this was an interesting study that even when it comes to symptomatic AEs, the current practice is that clinicians, are through interactions with the patients, collecting these symptomatic adverse events, but it assumes that they have an accurate picture of the patient's subjective experience. But this shows that although clinicians, have that good picture and have that expertise, it is really important for the patient to be able to also report how they're feeling. And yet clinicians systematically tend to downgrade the severity of symptomatic adverse events that the patients are experiencing. So patients, again, with these subjective experiences like pain and nausea and fatigue, they have the best representation of what that is and how it impacts their ability to function. And they are able to identify these symptoms earlier and rate them more accurately. And oftentimes they rate them more severely. And then on the right, you'll see that when we compare patient symptom reports to overall health status, it's actually the patient symptoms, patient-reported symptoms that are more correlated with overall health status compared to the clinician-reported symptoms. So again, just reflecting that it's important to understand from the patient's perspective, how they're feeling on the treatment. And the same we see for pediatric and caregiver reported symptoms too. So it's probably no surprise that a child's caregiver might have a better and more complete picture of their child's symptoms than clinicians do. So when looking at child reported and caregiver reported, symptomatic AEs on these graphs, again, you can see that the child and caregiver might have a better representation of their symptoms compared to the clinician's interpretation. And the child and caregivers report for more highly concordant with each other than the child and clinician reports. So, why use PROs to evaluate tolerability? This is kind of just a summary. We can get more precise data using tools like the PRO-CTC-AE. It provides more granularity of symptoms because the PRO-CTC-AE asks separate questions about frequency and severity and truly that interference with functioning. It can lead to reduced bias, more sensitive and accurate data. It's easier to collect more frequent data. So you don't have to rely on the patients reporting directly to the clinicians at site visits. It's an easier way that patients can self-report themselves, maybe at home every week, etc. Which then brings us to my next topic of what are the options then for PRO data collection. So of course, the most traditional is using paper, but while that's still an option, we're seeing this major shift away from paper and towards electronic data collection for a lot of reasons. More accurate data, better quality data, less missing data. I won't have time to go over all the advantages here, but we're really seeing this shift more to electronic data collection, and patients seem to also prefer electronic data collection as well. So there are options here: handheld device, tablet, bring your own device where the patient can use their own smartphone. And we're really seeing that this is a convenient and familiar experience for patients, and often one that's preferred by patients as well. We're also seeing kind of this superior measurement of quality of life assessment when we do shift towards the electronic collection, and that allows us to collect data more frequently. So this was a paper that came out last year that directly compared paper PROs that were collected just on the first day of each treatment cycle, which again has been the most common practice, with electronic PROs that were collected every 3 or 4 days at home. And importantly, there was high compliance across both groups, providing evidence that of the feasibility and good compliance that you see with at-home electronic PROs. And you can see that when you only collect PROs 1 day of the cycle, you miss that larger picture. So the times that patients reported higher symptoms and lower quality of life, these time points were completely missed if you only collected on-site paper PROs. And it's really that continual measurement of quality of life via that at-home electronic reporting that was able to give a more detailed, full picture of the patient's experience. So hopefully this was helpful for you all in thinking about how patient-reported outcomes fits into this picture of safety and tolerability in oncology trials. And hopefully I was able to convince you to think about how adding PROs can kind of enhance the assessment of tolerability, and then also the assessment of general symptoms and quality of life from patients too. So thank you so much for listening. And now I'll hand it back over to Sonia.
Well, thank you very much for that. I just want to say thank you very much to Kelly, Bob, Maury, and Todd for that very insightful presentation. And now I'd like to invite Maury, Bob, and Todd to put on their webcams as we begin the Q&A portion of the webinar. And I just want to remind our audience members that you can still send in your question using that questions window that's located on the right-hand side of your screen. Now, while you were speaking, we did receive a bunch of questions, so I'm now going to hand the mic over to Todd to moderate. So Todd, when you're ready, you may begin.
Great, thanks, Sonia. Well, I'm thrilled to see so many questions have come in throughout the session. We'll do our best to get to as many as we can right now, and certainly will follow up directly by email to anyone whose question's not answered real time here this morning. So with that said, let's jump right in. First question here will be directed to Bob. It's a question about sort of common pitfalls, it looks like. So what are some of the operational challenges you've commonly seen in assessing QT or cardiac safety, specifically in oncology studies, and how do you advise these best handled?
Thanks, Todd. There are a number of challenges that we frequently encounter in oncology trials related to QTc assessments. First, a lot of these patients are on many other meds, some of which may be QT prolonging. And many of them are started on QT prolonging meds during their treatment. For instance, many of the antiemetics that are used to treat patients in oncology studies also lengthen the QT interval. They may also develop hypokalemia or hypomagnesemia, which prolong the QT. And sometimes it can be very difficult in an individual patient to figure out, does the QT prolongation that we're observing represent an effect of the drug or not? So that's why we try and characterize the compound's effect on the drug over the entire population to give us a better idea of which patients need more frequent or closer monitoring.
Great, thanks. So this next one I'm going to send over to you, Maury. It's an imaging related two-part question, actually. I recall with the adriamycin cardiovascular toxicity guidances that MUGA used to be recommended as a modality for assessing ejection fraction. Why not use MUGA rather than echo? And then part two is, can you clarify when or what is the role for cardiac MRI as an imaging modality, which is considerably more expensive but less convenient for patients? So maybe you could start with the MUGA, and for those who may not be familiar, what is MUGA?
Yeah, so a MUGA is a gated blood pool scan where you take a radioactive isotope and you inject it, and it makes it pass through the heart first and then a second. Then we can measure the ejection fraction of both the right ventricle and the left ventricle, which is our primary concern for cardiotoxicity. The nice thing about it, it was relatively simple and reproducible. The challenge, though, is it's actually exposing patients to radiation. As you know, the #1 iatrogenic cause of cancer, which we're talking about today, is radiation. And so the last thing we want to do nowadays is to use modalities that radiate patients when it's not necessary. But I think more importantly, the echo just offers so much more value. You showed the ejection fraction, how the heart moves, the different walls. You can see the valves. You can look at the pericardium to see if there's fluid. You can even detect some early changes with the global longitudinal strain. So I think nowadays, not to say that MUGA can't be used, but in my opinion, I think echocardiography offers much more. Now, just to be clear about the second part of your question, the cardiac magnetic resonance imaging is the gold standard. It really accurately quantifies both the function, structure of the heart, but also the texture of the heart, where you can see inflammation such as myocarditis from the tyrosine kinase or checkpoint inhibitors that Dr. Kleiman mentioned. So I think my thought is that for routine monitoring, in patient care domain as well as in clinical research, echo is the workhorse, and MRI is used for further diagnosis, like the case I gave you today of my patient with amyloidosis. We could really see lots of scar directly inside the heart through the MRI that gave us greater certitude even before we had a biopsy of the patient's bone marrow. So I think MRI generally serves as a backup role, and in particular can be helpful if you're worried about myocarditis. It's the gold standard. We now think of cardiac MRI as sort of the imaging biopsy of the heart, where we used to stick needles inside the heart. Now we just take photographs with the MRI.
Okay, great. Real clear. Thanks. Also a two-part question here. This one is for Kelly. Striking the balance of not asking too much of a patient by adding multiple pros and potentially overburdening them with those surveys is always a concern. Have you looked into patient satisfaction and compliance rates with electronic surveys in oncology populations specifically? And then the second part was, do patients want to take assessments at home generally, or would it be easier for them to do so in the clinic? You sort of touched on that towards the end in terms of the risks of only doing in the clinic, but I think this is more sort of what a patient's want. So
yeah, it's an important question, because we do need to make sure we are striking the balance, and it's so important to keep burden low, and that's even something that's called out in the FDA pro guidance, to make sure that you're only asking what is needed and not overburdening the patient. So studies have shown that oncology patients do want to report their symptoms. They find it important. There is high motivation to do so. Clario also conducted a research survey study where we asked them how frequently they'd want to report their symptoms, and they do prefer kind of daily or weekly at-home reporting versus just at their at the site visit with their doctor, because they really do want to make sure that the doctors know how they're feeling. So they are motivated. We do see high compliance, but that doesn't mean they always want to complete pros in the moment. We really do need to make sure that we're striking that balance, and we're not just throwing the kitchen sink at them in terms of bureaus. So I would recommend, to, as was mentioned, kind of reduce repetition, be thoughtful about your pro strategy, making sure you're only including ones that are really relevant and not including ones that are maybe repetitive, and then consider options that are maybe easy or more conducive to their lifestyle. So maybe offering at-home data collection if that's what they want to do, something like a BYOD scenario, as I mentioned, making sure that they're comfortable with the device and maybe allowing them to use their own device to report their symptoms ad hoc. Making sure that you're just providing kind of easy, flexible solutions for them, I think is important.
Okay, great. Thanks. Really helpful response there. All right, well, again, thanks to the audience for joining us today and for all the engagement during the Q&A session here. I know we didn't get to most of your questions, but we do have your info, and we'll get back to you directly. Thanks also to the three panelists today for the super informative presentations. I'll turn back over to Sonia now to close us out.
Okay, well, thank you very much for those questions. We have reached the end of the Q&A portion of this webinar. If we couldn't attend to your questions, just like Todd mentioned, the team at Clario will follow up with you after this presentation. And if you have any further questions, please direct them to the email address that you see there on your screen, science@clario.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from X-Talks with access to the recorded archive of this event. A survey window will be popping up on your screen. Your participation is appreciated, as it will help us to improve on our further webinars. Now give me a few seconds. I'm about to send you a link in your chat box, and you'll be able to view the recording of this event with that link, and also share this link with your colleagues once they register for the recording as well. And get even more out of today's presentation by looking towards the materials that are available under the handouts tab, and we have one there for you. It's a PDF of this presentation, so I encourage you to do that. Now please join us in thanking our speakers, Kelly, Maury, Bob, and Todd, for that very insightful presentation, and for Todd being your moderator. And I just wanted to say one more thing. Bob will be back for another informative X-Talks webinar entitled CNS and Chronic Drug Trials, New Cardiac Safety Requirements. So mark that on your calendars, and we'll see you on Friday, May the 19th at 11 a.m. EDT. Okay, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, Kelly, Todd, Maury, Bob. Thank you. Bye, everyone.
Everyone, thanks for coming.
Thanks.